Fingerprints are the most infallible means of identification. They are considered to be the most important individual evidence. At the crime scene, we get invisible prints which are called latent fingerprints. These prints are hidden and they have to be developed by using one or more chemical reagents. Unfortunately, the prints which we get at the crime scene are weak, they are fragmented. We call them chance prints since they are obtained by chance. The criminal is very clever. He would take every precaution not to leave the fingerprints at the scene of crime. Now it all depends how we develop that fingerprint. The reagent has to be strong enough to react with the components of sweat so that sharp fingerprints are developed. Unfortunately, the concentration of chemicals in the sweat is too meager. Therefore, we have to develop fingerprints not only by virtue of chemicals but by virtue of other equipments also. One such equipment is the laser light. Laser, when it falls on some laser active dye, elicits fluorescence which makes the developed fingerprint more sharp, more enhanced. Some of the laser dyes are crystal violet, acridine orange, guanine green. When these are present in the fingerprint developing composition and the prints are developed, they may not be very prominent. They may still be weak prints, but then we can observe them under laser light. One of the most common lasers which are used for this purpose are the argon lasers. They emit blue-green light. This light is allowed to fall on the developed print. This is a sort of post-treatment method. But the light is not allowed to fall directly on the print. It is first allowed to pass through a lens. The lens serves two purposes. Firstly, it spreads out the light. It increases its area of exposure so that the entire print is covered by green light. The second advantage is that since the light is dispersed, its intensity is lowered. Otherwise, if it is allowed to fall directly on the fingerprint, it is likely to burn the surface itself. So low intensity light is allowed to pass through the prism or through the lens. The additional advantage of this technique is that the prints which develop, they are no doubt very sharp, but then the observer also has to be very careful. He observes it through a filter. Otherwise, if short wavelength light impinges on his eyes, they are likely to damage them. And the long wavelength light, that is harmless. So the person observes whether the fingerprint has been properly enhanced or not through the filter, and through the very same filter he tries to take a photograph. Normally very good photographs are obtained. The prints are clear, the prints are sharp, so sharp that sometimes even a lens is not required to observe them. You can compare them with naked eye itself.
After studying this module, you shall be able to know the significance of lasers in fingerprint detection. You'll learn application of lasers in enhancing developed fingerprints, and you'll also learn the importance of fluorescence in fingerprint detection. You know, laser detection of latent fingerprints is an extremely sensitive technique with a wide range of applicability. The methodology is based on the principle that a luminescent fingerprint impinged on a nanoluminescent surface produces laser excited fluorescence. The fluorescence so produced provides intrinsically such high sensitivity that fingerprints may be detected on difficult and unusual surfaces. The method also ensures detection of weak as well as old latent fingerprints. Laser technique is often used as a supplement to conventional procedures of fingerprint detection. Let us now have a look on application of lasers to fingerprint detection. The most common device used for the detection of latent fingerprints on difficult surfaces is the argon laser. The surface impinged with the finger mark is illuminated with a blue-green beam from the argon laser. Before it strikes the surface, the beam is made to pass through a lens. This serves two purposes. Firstly, it enlarges the area of coverage of the beam so that the whole fingerprint impression becomes exposed to the laser light. Secondly, the expanded beam protects the surface from burning. The imprint is observed through a safety filter. Such filters absorb the short wavelength components and transmit the longer ones, thus protecting the observer's eye. After the fingerprint is identified, it is photographed through the same filter. The identification of the fingerprint as well as its photography is carried out in a dark room. A flow diagram for laser detection of fingerprint is shown in the given figure. In order that light from the fingerprint ridges alone reaches the eye, the surface containing the impression should be non-fluorescent. If the surface itself is luminescent, then the ridge pattern is post-treated with a chemical reagent so as to generate the ridge fluorescence, which is either stronger than the background or of a different color. Alternately, the surface fluorescence may be blocked by using an appropriate filter. The use of lasers for fingerprint detection dates back to 1977 when it was reported that inherent luminescence of sweat deposit may be illuminated by an argon laser. No fingerprint treatment reagent was used in this investigation. The fact that fingerprint residue contains luminescers such as riboflavin and pyridoxine was considered as a possible route to fingerprint detection. However, the luminescent components in the sweat deposit are present in trace amounts and therefore the method had limited utility. It thus became evident that laser technique can be used for resolution of ridge pattern only after the latent imprint is given a post-treatment. Two broad post-treatment procedures may be used. Number 1, the latent fingerprint may be treated with a luminescent material. Number 2, the latent fingerprint may be treated with a chemical reagent which reacts with a sweat component to form a luminous reaction product. Now, let us have a look on post-treatment of developed fingerprints with luminescent materials. In this procedure, the finger impression is treated with a laser active substance which becomes physically adsorbed on the sweat residue. When illuminated with laser light, the ridge pattern produces a contrast via the background. Number 1 is powder method. The application of finely divided material and the subsequent removal of the excess powder by brushing, blowing, or tapping has been the universal method of intensifying fingerprints on non absorbent surfaces. Since the early days of fingerprint technology, the technique relies on the mechanical adherence of fingerprint powder to the moisture component of the skin ridge deposit. Conventional fingerprint powders consist of a resinous polymer for adhesion and a colorant for contrast. The adhesive gets adsorbed on the moisture component of the sweat residue, while the colorant gets adsorbed on the adhesive. In this manner, the ridge pattern is visualized. The commonly used adhesives are starch, choline, rosin, and silica gel. The colorant may be an inorganic salt like manganese dioxide or an organic derivative like eosin blue. A good number of powder formulations contain organic derivatives that fluoresce or phosphoresce upon exposure to laser light. Such compositions are useful for visualization of latent prints deposited on multicolored surfaces that would present a contrast problem if developed with conventional powders. Moreover, they can be used for developing weak prints. Some common organic compounds which have been used for preparing luminescent powders are acridine orange, acridine yellow, crystal violet, fluorescence, and rhodamine B. Luminescent powders containing lanthides complexes instead of organic derivatives have also been formulated. Lanthanide complexes offer several advantages, including benefits from large stoke shifts, long luminescence lifetimes, narrow emissions, ability of sequential assembly of complexes, and chemical variability of ligands. Moreover, such powders are suitable for detection of latent fingerprints on difficult surfaces such as wood, masking tape, and polythene. Another method is cyanoacrylate method. The principle underlying the cyanoacrylate method depends on the fact that when alkyl 2-cyanoacrylate reagent is allowed to vaporize, it undergoes polymerization. The polymerized ester has a tendency to get adsorbed on the sweat residue, imparting a white color to the rich pattern. The mechanism leading to the polymerization is depicted. You see, a china dish containing a few drops of cyanoacrylate and sodium hydroxide solution is placed in a fuming cupboard. The object bearing the latent prints is suspended from the roof of the cabinet. The item is exposed to cyanoacrylate vapors for about 2 hours until whitish colored fingerprint pattern develops. Even though cyanoacrylate fuming is a convenient and reliable method for detecting fingerprints, the developed imprints are white in color and therefore lack contrast. The contrast may be enhanced by post treating the cyanoacrylate developed fingerprints with a solution of luminescent dye. Such stains fluoresce upon exposure to laser light, revealing sharp fingerprints. When dye solutions alone are used for print processing, the results are not satisfactory. The reason being that the dye solution tends to wash off the sweat residue. However, when stains are used in conjunction with cyanoacrylate fuming, this problem is avoided. The cyanoacrylate polymer stabilizes the latent prints so that solution dye staining does not wash it away. Crystal violet improves the contrast of weak prints developed on polythene by cyanoacrylate method. The stain gets selectively adsorbed on the polymerized cyanoacrylate. Likewise, the rhodamine treated prints also get preferentially adhered to the polycyanoacrylate deposition and hence are suitable for laser examination. Moreover, the absorption spectrum of the stain matches with the blue-green illumination of the argon laser. Now, let me tell you something about post-treatment of developed fingerprints with reagents that form luminescent reaction products. The amino acids present in sweat deposition may be transformed into luminescent derivatives by treatment with a chemical reagent. The luminescent compounds, when examined under laser, reveals the rich pattern of the fingerprints. Number one would be ninhydrin method. Ninhydrin technique has traditionally been the most popular one for processing latent fingerprints on porous, absorbent surfaces like paper, cardboard, and wood. The method relies on the reaction of ninhydrin with amino acids of fingerprint residue. The reaction produces a purple colored compound called Rumen's purple, which becomes deposited along the ridge, making the prints visible. A 0.2 to 1.5% solution of ninhydrin in Freon 113 is sprayed on the exhibit from a distance of about 6 inches. After the solvent evaporates, the solution is resprayed. The surface is then heated to about 80 degrees Celsius without allowing it to come in contact with the heat source. Better results are obtained by steaming the article. For optimum development of fingerprints occurs at a relative humidity of 65 to 80%. The amino acids in the sweat residue neither interact with cellulose content of paper or wood, nor do they migrate within the capillaries of the substrate. Therefore, the ninhydrin method makes it possible to develop fingerprints that are many years old. However, since the concentration of amino acids in perspiration is usually quite low, the developed prints normally do not show a sharp contrast. In order to improve the working performance of ninhydrin reagent, the developed prints are post-treated with group 12 metal salts. This interaction converts a non-fluorescent luminous purple into a photoluminescent complex. The fingerprints developed by ninhydrin zinc chloride method become highly fluorescent under argon laser light. The 480 nanometer line of argon laser is appropriate for excitation of the orange zinc luminous purple complex that absorbs at 485 nanometer and emits at 560 nanometer. As a result, a pronounced improvement in their detectability is observed. Moreover, with zinc chloride post-treatment and argon laser examination, the ninhydrin method may be extended to the detection of latent fingerprints on non-porous surfaces like glass and stainless steel. The complexes formed by the combination of Reuman's purple with europium and terbium salts too have been found suitable for fingerprint development, particularly on fluorescent surfaces. These complexes show enhancement of lanthanide luminescence via intramolecular energy transfer. Moreover, the lifetime of the luminescence is much longer than that of the usual background fluorescence. The lanthanide complexes are therefore suitable for time resolved luminescence imaging. Next is fluoroscamine method. Like ninhydrin, the fluoroscamine reagent too reacts with the amino acid content of sweat residue, producing luminescent reaction products which render visibility to finger marks. A solution of 20 mg fluoroscamine and 0.5 ml triethylamine in 100 ml acetone is sprayed on the surface containing their finger mark. The surface is dried and observed under the argon laser. Experience has shown that fluoroscamine not only possesses a great sensitivity than ninhydrin but also works well for the detection of latent fingerprints on multicolored surfaces. Dear students, now let us summarize this chapter. So, in this chapter, we have learned that the strong fluorescent produced in a post treated finger mark with an argon laser enables detection of imprints on unusual surfaces. It also enables detection of very old fingerprints. The prints may be processed at room temperature. An arc lamp too produces fluorescence but only when the sample is plunged into liquid nitrogen and illuminated at minus 200 degrees Celsius. Thus, larger articles cannot be examined by the arc lamp method. The size and shape of the exhibit does not matter when illumination has to be carried out by a laser. There are a few disadvantages associated with laser. The casework has to be performed at the fingerprint bureau. Portable lasers, which may be carried to the scene of crime, are not yet available. And then we have learned that laser equipment is quite costly. When optical spectroscopic techniques are used in combination with lasers, the operational procedures become tedious and complex. And lastly, we have learned that however the end results obtained with lasers are so remarkable that one becomes inclined to believe that their merits outweigh the demerits.